Our project is a little bit more focused on creating tools to infer phylogenies, as the title of our group kind of implies. There's me, Alex, Christian, Dongwook, Jamie, Caitlin. And we started with motivating questions about the relationships between lots of different parts of the virosphere that look like they might share folds or share an evolutionary history, but the phylogenetic signal that kind of ties them together is lost, and we can't really use amino acids to reconstruct that history. So if we use protein structures, we can stretch typical phylogenetics a little bit further, and that's what motivated this project. The fact that viruses evolve extremely fast, using structure kind of calms that down and lets us do phylogenetics a little bit deeper than we usually would. And we have a lot of these open questions. It's also a motivation on the NIH side to describe some clades which have very high pandemic potential. We don't really understand their evolutionary dynamics yet. And one of these clades, flaviviruses, has been described in a paper where they do exactly this. They use structural phylogenetics in order to pin down what exactly these different flaviviral proteomes share. So, as we know, viruses like to mix and match. There's a lot of lateral transfer. So they wanted to see what all these flaviviruses held in common as far as their folds, as well as study the evolution of each fold and the global flavivirus landscape and get a good tree that represents the whole thing. So they struggled through using ESM and AlphaFold to create a foldome map of all of these different viruses. What we want to do is avoid that heavy compute to get all of these structures. What we'd like to do is just predict 3DI characters to allow us to align these things coarsely without actually knowing the underlying structures. And 3DI characters basically represent local structural motifs. So yeah, we want a model that performs well with viral proteins. So what we're going to do is use some pre-existing tools and adapt them. So here we have the PROS-T5 large language model, protein language model, which is able to output structural tokens or local structural motif tokens. Fold tree, which is used to align these things and create phylogenetic trees, and Foldseek and Unicore, which are tools that are used to find shared core genes between the viral proteomes. So we're going to tune all of this pipeline for creating structural representations of amino acid sequences, finding core genes, and making trees towards viruses. The data set we used was BFVD, so a big friendly viral data set. And here we have basically an effort to create a viral foldome map of everything in the UniProt database, since it wasn't available in AlphaFold DB. So this is just kind of representative snapshot of everything that's viral in UniProt. We're going to use the PROS-T5 and fine tune this in order to predict the local structural motifs of all of these structures. So we're using LoRA to fine tune this. Here's just a small little data set to test the fine-tuning process, which is actually still ongoing right now. So this model is being trained to predict basically the local structural motif logic of viral sequences as we speak. And to accommodate this model, instead of using the typical fold tree pipeline, I adapted it to use the LLM instead of the structural tokens coming from actual structures. So this tool is actually working, and you can just input amino acid sequences and it will derive a tree using what the LLM guesses should be the structural motif at each position. We've compiled a preliminary data set of viral proteomes, and we're going to try to make species trees and core gene trees for each of these viral proteomes. So here we've tried to get some core genes across these viral proteomes using the naive model that hasn't been fine-tuned. So we're already seeing a pretty decent quantity of core genes across different viral clades that are very wide. So you wouldn't expect to find these necessarily consistently with just amino acids. And we've already started creating trees with the naive model, so that also works. And we're going to keep working on this project because these are open research questions that our group has, and we think there's kind of a gap there in viral evolution as evidenced by the paper on flaviviruses. So yeah, some limitations. You wouldn't use this for closely related viral strains. This isn't for an outbreak. This is studying deep viral evolution. And we've had some trouble integrating these fine-tuned models into the existing stack of tools. So these are based on C++, and you have to compile a model in a very weird way. And yeah, we kind of ran into trouble with that. But we're getting close to having a working tool stack and a fine-tuned model. So pretty soon we'll be exploring the biosphere and hopefully coming back with some insights on how these things evolved.
Thank you.